video, you're gonna learn about um, the structure of the monoclonal antibody, and then this is the monoclonal antibody. Um, so you're gonna learn about this Fab and the FC region of the monoclonal antibody, and then what are the functions of Fab and the FC region. So the monoclonal antibody looks like a Y-shaped antibody, as you can see, and then this region from here to here is called FC region, and then this region is called Fab region. So I'm gonna go into detail about what each region does, but just to explain you a little bit more, so there is a heavy region and then heavy chain, and there is a light chain. So of this segment and this green segment and this blue segment is heavy. It's all heavy, and then this dotted part is the light chain, and this fragment is variable, and this one is constant. And then it has a CDR loop, which is complementary determining region. So each segment, variable segment, and the light chain segment and the heavy chain segment, the variable part of the antibody has uh, CDR loops embedded into the segment. So in order to give you a clear picture of how it looks like, so this CDR2, CDR1, and CDR3. This is how it looks like, and here it is. The heavy chain has CDR1, CDR2, CDR3, and also the light fragment has CDR1, CDR2, CDR3. So as you can see in this picture, this is CH1, CH2, CH3. This is all constant. This is heavy part. This is light, light chain, and it has VL. The first fully human antibody was introduced in the market with significant success was Humira using the phage display approach. The heavy chains consist of three constant region, CH1 to 3 as we discussed, and are connected through disulfide bond. And both light and heavy chains have a variable domain, VL and VH respectively. Each pair forms one of the two variables fragments, FV of an uh, IgG antibody. The six hypervariable CDR loops L1 to L3 and H1 to H3 embedded in the heavy and light chain variable domains form the two antigen binding sites which are identical in normal antibody. So the three CDRs, CDR1, CDR2, and CDR3, arranged non-consecutively on the amino acid sequence of a variable domain of an antigen receptor. So each light fragment and heavy fragment has CDR1, CDR2, and CDR3. So the six hypervariable CDR loops L1 to L3 and H1 to H3 embedded in the heavy light chain variable domains form the two antigen binding sites which are identical in a normal antibody. So let's get into different function of different parts of the antibody. So as you know, there's a Fab region. So the function of the Fab region is antigen recognition, and one of the major functions of the Fab region is antigen recognition, and then the neutralization of pathogens. Once the antibodies recognize the antigens, the binding occurs outside the cells. This is where most of the bacteria and bacterial toxins are found. The binding prevents the access of the pathogen into the cells and prevents infection or destruction of host cells. So Fab is basically recognition. I'm gonna highlight this mostly for recognition and neutralization of pathogens. Uh, but the FC mediated function is the activation of effector cells, and so through their FC fragment. So this is very important. I'm gonna put it right here. So these are the three functions. Um, so the first one is activation of effector cells. Through their FC fragments, antibodies can activate accessory elector cells. So elector cells include phagocytic cells like macrophages and neutrophils, T cells like natural killer cells, and eosinophils and mast cells. Each of these cells has a receptor for the Fc fragment. Thus, these cells can identify an Fc fragment and eliminate the pathogens. So I'm going to show you a picture. So here you can see this is the antigen. These are the antibodies, and this region, as you know, is the Fc region. And once the Fc receptors recognize cell bonding antibodies, uh, then this cross-linking of CD16 trigger degranulation into a lytic synapse, and then this tumor cell die by apoptosis. So this is antibody. These are the effector cells. So Fc region is responsible to activate the effector cells and the opsonization. Those microbes that replicate outside cells are removed by an interaction of the Fc part uh, on the surface of elector cells. The antibodies coat the surface of the pathogen and allow binding of their Fc domain to Fc receptors present on elector cells. And then complement binding. Once bound to the antigen, there is formation of antigen antibody complexes. This further activates a complex set of reaction called the complement cascade. So in conclusion, Fab region is only responsible for antigen recognition, and Fc region is responsible and also the neutralization of pathogens. So neutralization of pathogen is also very important. So once the antibody recognize the antigen, um, the binding occurs outside the cells. This is where most of the bacteria and bacterial toxins are found. The binding prevents the access of pathogens into the cells and prevents infection or destruction of host cells. And so it actually blocks the binding of the bacteria to the host cells.